Hello, everyone, and welcome to this Balchem webinar. My name is Stefano Vandoni, EMEA Technical Manager for Balchem. Bioavailability of rumen-protected products is one of the key aspects when defining the cost-benefit ratio of these nutrients. While for other nutrients bioavailability estimation is not an issue, unfortunately, there is still a lot of confusion relative to rumen-protected choline. For today's Real Science webinar, we welcome Dr. Joe McFadden. Dr. McFadden will try to help us make some more clarity about the definition of bioavailability with particular focus on rumen-protected choline in dairy cow. Dr. Joseph McFadden is an associate professor in dairy cattle biology at the Department of Animal Science at Cornell University. He received his BS with distinction in research from Cornell University in 2003, his MS from the University of Illinois at Urbana-Champaign in 2005, and his PhD from Virginia Tech in 2009. Dr. McFadden started at Cornell University in 2017. His research interests center on the study of lipid nutrition and metabolism in dairy cattle. Joe, the floor is now yours.
Thank you, everybody, for joining. Good afternoon. I'm going to talk about choline bioavailability and the challenges that exist to actually attempt to quantify this. All right, so we're going to go through the definition of what bioavailability actually means. I think it's a term that is probably one of the most misused terms when we talk about rumen-protected supplements. So we want to be really clear about what bioavailability actually means. Discuss some established knowledge about sort of choline biology and choline nutrition. I'm going to introduce a couple new concepts that we're working on that will hopefully help us better understand how choline is degraded in the gastrointestinal tract, which will be important if we want to eventually define metabolizable choline requirements. And finish with discussing what types of methodologies exist and what are their limitations to measure choline bioavailability, and finish with just a couple slides to emphasize that biological outcomes matter when it comes to choline bioavailability, and that we want to see increases in milk production, improvements in animal health, and highlight some new findings that are out there that emphasize the amazing things that choline can do. So I actually took this paper. This was a paper from Godber 1990. It wasn't talking about choline in this paper, but they were talking about what bioavailability actually means and how do you define it. And so I took the table and the terminology they were using, and I used it to describe choline bioavailability. First, you obviously are feeding choline. We're feeding it as a rumen-protected supplement. This is obviously why we're having this conversation, but at the same time, there's choline as a natural sort of nutrient and feed, and that's something that's often ignored, but it is certainly part of the diet. And so when we look at abundance of choline in the diet, it's part of choline in feed, but also rumen-protected choline. Digestibility, that refers to the amount of choline that escapes rumen fermentation and released from encapsulation by the digestive process. When we look at a lot of different in vitro techniques that we're going to talk about, this is what we're trying to measure, digestibility, choline digestibility using in vitro methods. Absorbability, or I'd rather use the term metabolizable, refers to the amount of choline that is released from encapsulation, or the amount of choline that gets to the intestine that is actually absorbed into the cow. And bioavailability refers to the amount of choline which was absorbed but utilized by the cow for a biological function. I don't think that we have too much to worry about in terms of choline, but there are certainly some nutrients that could end up in urine, and that would not represent a bioavailability. And so with regards to choline nutrition, we're thinking about how much of that choline is being used to actually support a biological function like milk production. Now, what bioavailability is not, it is not the amount of choline that escapes ruminal fermentation. And so simply doing an in situ incubation in some rumen fluid and measuring the amount of choline that exists post ruminal degradation, that is not bioavailability. Right, that is a measurement of rumen protection. But you could certainly have a scenario where A rumen protected supplement could be overprotected, could have great rumen protection, but not be released in the small intestine and end up in the feces. All right. 2, the amount of choline not measured in feces. And what I mean is that you could certainly measure the amount of choline from a rumen protected supplement that ends up in feces and look at the difference and say, well, that difference was what was released in the cow and that was bioavailable. That's not accurate, because certainly if it's degraded in the rumen, that's not going to contribute to bioavailability. And there's actually evidence now that there could be some post-ruminal GI degradation of choline. That all collectively is going to limit absorption of choline. The amount of choline measured at the small intestine, again, just measuring it at the small intestine, that would not be bioavailability, because we need it to be absorbed and used for biological function. And again, the amount of choline not used for biological functions. So we want to think about the long-term outcomes here when we talk about bioavailability. Now, choline can do so many different things. This is probably the most complex slide I have. We wrote a review paper about this 2 years ago, about the role of methyl donors. But, you know, choline is a complex nutrient, because it's not like methionine, where you can go and measure milk protein. You can measure methionine content in milk, methionine in plasma. You know, choline immediately, once it's in the GI tract, it's going to start to be metabolized. Once it gets into the gut tissue, it's going to be metabolized, and it can be metabolized differently throughout the body. And so we tend to talk not just about choline, but choline metabolites. And so some of those are in blue, like betaine, complex phospholipids, like phosphatidylcholine. And that's one that's really important to make lipoproteins in the liver and help that transition cow dispose of fat during that transition period. Phosphocholine, that's actually a key nutrient in milk, especially in early lactation. You'll see high concentrations of phosphocholine. Now, what really complicates this, and this is not the right place to go into this, but there are certainly different pathways that use this. If you just draw your attention to the methionine cycle, you know, we tend to define choline as a methyl donor. You know, choline has 3 methyl groups. It can donate those methyl groups to the cycle, contribute to methionine synthesis, but also S-adenosine methionine, that's SAM, and that's a universal methyl donor. And in the past decade, there's been so much research showing the importance of methyl donors, but also S-adenosine methionine for methylation reactions. And this process is really important to make complex phospholipids to help that cow dispose of liver triglyceride. There is an alternative pathway, however, that just uses choline itself, doesn't require methionine. And so there's often a lot of debate about choline and methionine and which one is better. And well, I like to say, well, they're 2 very unique nutrients. They have 2 very unique functions, and you can't replace one with the other. And this is a perfect example that in this pathway, the CDP choline pathway, you can only use choline. There's no role for methionine there. Now, fortunately for us, there's been a lot of research focused on choline nutrition over the years. You know, this is a paper in 2016 that was just measuring the amount of choline and choline metabolites in various feed ingredients. And so just emphasizing that the cow already has choline in her diet. Obviously, this is not protected, but it is in various forms in various feedstuffs. But the one thing that they discovered very early on in the 80s was that if you give either these different feed ingredients that have choline or choline chloride, which is at the bottom, this is what's being protected, it is rapidly degraded by ruminal bacteria. Right? And so this was an in vitro experiment where they had different incubation times of 15 minutes to 48 hours. And the higher this number represents the extensive ruminal degradation of choline. And so you can see within 15 minutes here in this measurement, they had 97% disappearance of choline after 15 minutes, which is quite remarkable. It takes a little longer for choline in feeds, but still quite extensive degradation. And so there was a lot of interest to be able to consider how do you increase post-ruminal choline availability to enhance the bioavailability of this nutrient. And one of the earlier studies was this one, where they abomasally infused choline at different doses, relatively high doses compared to what we're currently feeding. But the really important point here is that in a lactating dairy cow, if you can get the choline post-ruminally to the small intestine, it's going to be absorbed and it's going to increase milk choline concentrations. And so there's often some debate about whether you should see a choline response to rumen-protected choline. When you look at the Abomasal infusion studies where they abomasally infuse unprotected choline chloride, I've yet to see a study that does not see a choline response, either in plasma or milk. And so that suggests that if you have adequate intestinal release of choline, that is going to be bioavailable to serve biological functions like milk production. So I've tried to, this is not in a formal paper or anything, but I had my graduate students just start to collect studies that have been performed over the years that looked at the choline response or choline metabolite response in various studies that investigated either Abomasal choline chloride infusion or dietary rumen-protected choline supplementation. And they looked at these various studies that looked at milk or plasma samples for these different metabolites. And I just had her go in and say, okay, if there was a major increase, 2 pluses, a minor increase, small plus, if there's anything negative, put negative, and NC, meaning no change, that there was no difference. And so when you see something like this, it really says, okay, and you look at the totality of the experiments, whenever you're going to abomasally infuse or feed rumen-protected choline, the general consensus is that there's going to be an increase in either milk or plasma choline, betaine, and phosphocholine. Okay. Now, something to keep in mind is that the analytical technologies to measure these things really didn't become accessible and affordable until the last 20 years, 10, 15 years. And so I expect that over time, we'll be able to see many more of these metabolites. But the one thing that was pretty obvious when you look at this table is that when you look at the complex phospholipids that have the choline moiety, for the most part, you don't see any response. You do see a response in dry cows, but for lactating cows, you don't. So I think this is important as we move forward and we want to define whether or not there was absorption. We certainly can focus our attention on choline, betaine, and phosphocholine as being probably the top 3 nutrients that are metabolites that are going to be responsive to a rumen-protected choline feeding approach. So one of the things, though, that I've focused my research program on in the past few years was on how is choline degraded in the GI tract, and how does that potentially influence bioavailability? And there's been an emerging metabolite called trimethylamine oxide. Certainly, it's been around for millennia, but our focus on TMAO, which is how it is defined, is a relatively recent occurrence. And what's going on here is that bacteria have enzymes. Those enzymes are called TMA lyase, and there's different types of them. But they'll take methylated amines like choline and degrade it to trimethylamine. This has been known for some time that, rumen bacteria have this. I mean, this is how choline is degraded in the rumen. That trimethylamine can be used a little bit to make methane, but we have evidence to suggest that it's also absorbed across the rumen epithelium, and that increases TMAO synthesis. And that TMAO not really metabolized in mammals, but it ends up in urine and a little bit in milk. And things that we appreciate now is that there's certain types of microbial phenotypes that are going to drive this degradation over others. We don't really know much about that in the cow, however. Now, Rich Erdman did a study very early on just showing that in rumen fluid, here's the choline degradation that you have, and this is what you would have if you do not have protected choline. Rumen-protected choline chloride wouldn't do this. But if you had unprotected choline chloride or choline in feed, this is what happens, is that it gets converted to trimethylamine. This is avoided, though, with rumen-protected technologies. In panels B and C, though, this is what sort of kicked off this interest, is that we abomasally infused choline chloride. So we bypassed the rumen. And when we did that, we increased plasma concentrations of choline, but we noticed that there was a marked increase in circulating TMAO concentrations. So this is the first evidence that there was post-ruminal degradation of choline to trimethylamine, and that eventually would get absorbed and be made into TMAO. I don't know the extent of that. It could be, if you look at non-ruminants, it's quite, they estimate that it's around maybe 10% that gets degraded to trimethylamine. In the cow, I just don't know, but it is a factor that it's going to influence bioavailability. And I'm going to come back to this because when you look at in vitro methodologies that don't account for intestinal bacteria or this intestinal trimethylamine lyase, they are overestimating bioavailability. Or I wouldn't even call it bioavailability. They're overestimating what's going to be available for metabolizable choline. We went and did a study too where we gave, this is not specific for choline. We actually gave lecithin. Lecithin has choline in it as a phospholipid. And we actually saw increases in plasma TMAO as we increased the dietary intake of lecithin. And so obviously this was not protected lecithin. This was also subjected to ruminal degradation, but same principle. This is just showing you the importance of the microbiota, and that this was actually some rodent studies where they had pre-antibiotics, and so lots of the bacteria were intact, full microbiome. As choline starts to go down, you can see TMAO go up, and the conversion of choline to betaine starts to go down, just showing you that degradation. But if you had antibiotic treatment, that was able to suppress TMA lyase, and that was able to maximize choline bioavailability. So I think there's opportunities for us to think about how to do this through nutrition. There are potentially nutritional approaches to reduce choline degradation. This is receiving a lot of interest, though, in the human world. I'm not going to spend too much time on this, but, in vegetarians and omnivores, they were able to show that meat eaters, which consume a lot of carnitine, they actually have higher plasma TMAO. And this is because they have a microbiome that has a high TMA lyase expression. And so we have early preliminary evidence suggests that there are scenarios that could potentially influence TMA lyase expression, and they're therefore influenced by availability of choline. And so far, this has been ignored. And there's also a lot of individual variation they've documented in the literature. And this is actually humans that were given 0 to 6 egg yolks, eggs having high phosphatidylcholine. And they were able to show that in response to that, for example, human number 5 really didn't respond at all. There was no increase in TMAO, but human number 3 had a very high response. And so human number 3 had a high choline degradation, whereas number 5 didn't. And that was most likely had to be due to the fact that they had a very unique microbiome. And there's plenty of research to show now that microbiomes, even though they're fed the same diet, they could be highly variable across a herd. And so we have to be very aware of when we're just doing studies that are using rumen fluid, maybe from 1 cow or 2 cow, there's a lot of potential overinterpretation when we do that because of variable microbiomes. Now, just so we wanted to actually study what the TMAO could be doing, and we've only done one study like this. And if you look at the non-ruminant literature, there's a lot of talk about how TMAO can modify metabolism. Well, we did an intravenous infusion study. These were early lactation cows. We increased circulating TMAO levels. As you would expect, they had more urine TMAO yields and milk TMAO yields. I'm not going to get into the biological outcomes because there weren't any. It didn't impact milk production. It didn't impact measures of metabolic health like glutathione NEFAs or insulin. It didn't do anything, which is a good thing. But what's not a good thing is that that any choline that's converted to TMAO means less choline bioavailability. But the one thing I think is important here is that you can measure TMAO in milk. And I think this could be a potential tool moving forward to compare different types of rumen-protected choline as a way to look at relative bioavailability. So what are those methods to measure bioavailability? Well, one method that we've recently used that will hopefully be published very soon is where we look at the temporal changes in plasma and milk choline and choline metabolites in response to increasing doses of rumen-protected choline. All right, and so this is sort of a bolus administration. We do a sort of a dose titration curve, a single bolus, and then we look at the plasma and milk metabolite response over time. We realize that there's no real perfect method to try to attempt to measure bioavailability. Certainly, this one does not mimic feeding behavior, does not allow for mastication by the cow, perhaps. It's qualitative, not quantitative. And it's certainly with a single bolus, you can't expect to see many biological outcomes. But you can certainly look for choline content in milk. And I would call that a biological outcome because that is milk synthesis. And so, but it does allow for a good approach to compare different products. And so we started to explore this. For example, here we have a scenario where we gave a rumen-protected choline bolus, and we were able to show an increase in plasma choline response to that bolus, as well as a dose response in plasma betaine with increasing doses of choline chloride. We've done this study with giving choline chloride as a rumen-protected form inside of a gel cap, but we've also given it as a TMR. So it's a mini TMR meal. You put it in front of the cow, she chews it, and we go ahead and we do this temporal plasma response. We also look at milk as well, and in this particular rumen-protected choline we were studying, we saw an increase in choline yield in response to increasing doses of choline chloride as a rumen-protected supplement. And so I want to make it clear that this was actually in one milking just 8 hours after administration of choline chloride. And so it was quick. You went ahead, we gave it, and then the 8-hour mark and the 16-hour mark milking post-bolus, there was an increase in choline yield in milk. So we actually looked at the bottom two studies. These were some of the other metabolites that we looked at in these studies. And with this bolus approach, we did see, again, an increase in choline, betaine, and phosphocholine in one study, and choline and betaine in another study. I'm not going to get into the nuances of those studies now, but hopefully that paper will be published soon, and you'll be able to see that. Now, the one thing that we did learn, though, from this study is that we tested different types of prototypes. Now, these prototypes had different concentrations of choline chloride in it. It ranged anywhere from 30% to 60%. Overall, we tested 5 different rumen-protected choline prototypes. And this was probably the most pronounced response that we observed. Now, the same thing happened with, well, I'll show you in milk in a second, but there's 2 things I want to point out. And the first one is that look how fast the plasma TMAO response goes up. That in prototype 1, it's 2 hours, 2 hours, and 2 hours for all 3 of those prototypes. And it's so quick that this has to be a reflection of ruminal degradation of choline. That we all know that there's no perfect rumen-protected supplement out there, that there's going to be a small amount of release. And in fact, I would argue that you want a small amount of ruminal release, because if you had none at all, that would be a good indication that you had overprotection, and there's probably not going to be intestinal release. All right. And so it's a fine line to be able to have just enough protection, but not too much, and so that there's intestinal release. Now, keep in mind that in the previous figures, we saw the betaine choline response in that 10, 12, 14, 16-hour range. And that would be analogous to when that rumen-protected choline supplement got to the small intestine. We do not see a TMAO response here. And so with these particular prototypes, that's a good indication that there was not extensive post-ruminal choline degradation to trimethylamine. Right? But the other thing I want to point out here is probably one of the most important things of this slide. Is that look at the variation. Keep in mind, we gave an equivalent dose of choline chloride, 0, 18, 36, or 54 grams. But the prototypes were just different, meaning they had different amounts of protection. But the TMAO response was noticeably different between prototype 1 and 2 and between 3 and 2. And so this, again, could be a good sort of marker of choline degradation. Interestingly enough, the milk response followed the plasma response, that whenever we had the highest plasma TMAO, which was prototype 2, it also ended up in milk. And so this is, again, a good I think, marker of gastrointestinal choline degradation. One thing we wanted to point out, though, that in this study is that we really haven't done the right study. This is only 12 cows, mind you, but we took the top and bottom quartile of those 12 cows, so the bottom 3 and the top 3. We had different prototypes here, P1 through P3, and looked at that area under the curve for that TMAO response. And there was about a twofold difference. And so this says that there are certain cows that are going to have a higher TMAO response than other portion of cows. That means that some cows are going to have a higher bioavailability of choline than others. The microbiome has to be most definitely influencing that, but we really don't have a firm understanding of the degree of that influence. Now, certainly, an alternative method to measure bioavailability is this one. This was looking at the Abomasal infusion of choline chloride versus dietary rumen-protected choline as a top dress. This was most recent in 2016. This is a very intensive approach. I want to keep in mind that in this particular study, the bioavailability of the choline chloride that was infused was actually estimated to be around 60%. And that was unprotected, mind you. And so just because you have unprotected choline chloride getting to the intestine, you're not necessarily going to get 100% absorption. And that's the data suggests that they're not. And so what is that difference? What is explaining that 40% difference? And perhaps it is the conversion of choline to TMA, but they also cannot quantify the role the GI tract plays in metabolizing choline. And that's a very difficult thing to do, certainly in a cow, an expensive thing to do. But it's important to realize that not all of the choline is going to be bioavailable. Now, certainly with the rumen-protected version, there was less bioavailability in this particular study. But we want to point out too here, though, that this does not count for, again, the conversion of choline to TMA. It can be influenced by stage of lactation and feed intake, and the rate of the digestive flow can certainly vary and impact study outcomes. Another approach, and this is real funny, I'm not going to get into why it's funny, but we actually been recently encouraged to consider the Ross method. Well, the Ross method is an in vitro method. Some of you may know what this is, created by Debbie Ross and Mike Van Amberg, my colleague across the hall. And they have these, also we have 3-step in vitro procedures as well. But I would just say in vitro techniques as a whole have limitations, right? You have in situ or in vitro rumen incubation times that are fixed. You're really not controlling for particle size influence, especially with that Ross method. The digestive enzyme cocktails that are unknown or based on sheep studies. Really, these methods were not developed for rumen-protected fat-coated supplements, and so that requires adequate peer review. Use of bags prolongs lag phase of digestion. There's a demonstrated loss of highly soluble components of feeds. And the one thing I've been advocating a lot for is that this does not account for that post-ruminal conversion of choline to TMA. And if you look at that last paper by DeVeth, I think you got to pay attention to it because there's a 40% we can't account for. And so we need to think about what role that could be playing, and the in vitro methods don't count for that. And so they're most certainly overestimating by what's metabolizable, potentially bioavailable, but we just don't know the degree of that overestimation just yet. Does not account for effective product handling or mastication by cow. Something we thought about in our bolus approach was in our second study, we actually gave it as a pre-meal. They were able to chew it. start to break it apart. We actually even preconditioned those prototypes with ground corn. So we mixed it with some ground corn. You could do it with different types of ingredients, but the idea meaning that the degradation of a rumen protected supplement doesn't start once it's sort of manufactured. It does start as soon as it's manufactured, but you have to account for handling. You have to account for how the cow uses it, the external factors, the factors of the environment, and so we have to put more thought into that. Most importantly, these in vitro methods do not measure bioavailability. I've had some smart people try to tell me that's the case. That is not the case, okay? It is not. I will not agree with that. I'll leave it at that. Rumen bypass followed by duodenal insertion. So if you do an in situ incubation, maybe it's for, 8 hours, 12 hours, 16 hours, and then you take it out and then you insert it into a duodenal cannula and then you collect it in the feces. A lot of the same limitations. I would argue, in addition, that the intestinal insertion likely does not align with pancreatic secretions, and it falsely assumes that choline release in the large intestine is bioavailable. And what I mean is that it's going through that entire intestine. We just do not know how much choline is absorbed in the large intestine. And if you were to look at the sort of expression of choline transporters in the non-ruminant literature, it would argue that it's probably fairly minimal, but it potentially could be released in the large intestine. There's bacteria in the large intestine that could be influencing its degradation. So just measuring it in feces and saying, okay, well, the difference that we don't see in the feces, that had to have been bioavailable, that is grossly inaccurate. Okay, we should not say that. We can call it choline release. I would not even call it metabolizable choline because we don't know the role of post-ruminal bacteria. So there are other approaches like rumen-protected choline feeding trials, obviously. This plasma-free amino acid dose response technique, I say amino acid because this actually hasn't been done yet, as far as I know, with choline. But colleagues at the University of New Hampshire have done a lot of this work, and as well as some labs across Europe. And, perfect world, you stable labeled choline, that's what's done in non-ruminant sort of studies to be able to trace its utilization, but that would be very expensive. I want to finish by saying that bioavailability at the end of the day comes down to biological outcomes. And the one thing you have with rumen-protected choline feeding over the years are, I think there's 3 meta-analyses. I could be wrong. Maybe there's 4 now. There's a lot in the past couple of years. But these meta-analyses all tell the same story. We don't really need to question it, I don't think. And that same story is that rumen-protected choline feeding increases milk yield. At one time, when I was in graduate school, it was you got to feed choline to prevent fatty liver and ketosis, which I'm not going to argue with that. But then it was like, okay, you got to feed rumen-protected choline in the transition cow to increase milk production during early lactation. Well, now there's enough studies to show that you could feed rumen-protected choline in mid-lactation and late lactation and still have a beneficial outcome on milk production. That I think we need to open up our eyes to that. Now, one of the things that they've shown repeatedly is that there is modest increases in intake when you feed rumen-protected choline, but it doesn't explain everything. It's estimated to explain about half of the increases in milk production. And so what happened to that other half? Well, choline's probably doing things in the mammary gland, mammary tissue, to enhance milk synthesis. Perhaps choline is enhancing lipid transport from the liver to bring more nutrients to the mammary gland to support milk production. I mean, we really need to get a better understanding of that for sure. And one of the things that other people have realized is that choline works to increase milk production at varying levels of dietary methionine status. And so here's looking at metabolizable methionine on the low end, 1.65%, on the high end, 2.65% of metabolizable protein. And you sort of have to look at this figure bottom to top, and you show that incremental increase in milk yield is greater in diets that are low in metabolizable methionine. You can still see increases in milk yield, just not as pronounced, though, in diets that are higher in metabolizable methionine. So my final slide here is that there's really a frontier that I call it the next frontier of choline biology, And it's because of all the great research that's being done across a bunch of different labs. Some things I stumbled upon that I thought were really interesting. This was actually in some non-ruminants, but they showed that endotoxin or lipopolysaccharide enhances choline uptake to enhance macrophage activation, which could be really important, right, for transition cows. It could be important potentially for heat stress cows. And so there's been actually work by Dr. Barry Bradford. We have some work coming out too showing that choline or lipids with the choline moiety are able to influence immune function. And so I think there's going to be a lot more on that coming forward. There's been work looking at heat stress, 2 studies. The second bullet was an in vitro study looking at mammary epithelial cells. They basically exposed those mammary epithelial cells to heat stress environment, promoted oxidative stress, it killed the cells, but when you gave them choline, it prevented all that. Dr. Heather White did a recent study where she heat stressed some dairy cows, and there was a tendency for improved milk production. They also had improved clinical health, so lower respiration rates. And finally, choline acts during pre-implantation development of the bovine embryo, the program postnatal growth. Oh, this is super exciting work at the University of Florida, showing that when they did this, there was accelerated growth in the offspring. So we need to think more about the sort of choline within that regard. I'd like to thank my lab. My current lab is here. Actually, it's not my current lab. I should have updated the slide with a couple new students. But Tanya France, William Myers, and Crystal Chang played a key role in some of our choline-related work. And you can see my collaborators on the right-hand side. I'd like to acknowledge our major support. You know, we get some funding from NSF, USDA, FAR, that certainly support students, but I'm going to acknowledge support from Volchem, Northeast Agribusiness and Feed Alliance, and the Hatch Formula Funds as well. I'll take any questions if there are any.
I think some of them has been already answered during the presentation, but I'm going to ask them anyhow because maybe we can go a little bit deeper on some of them. So there has been one question about a study that was using mobile bag technique to attempt to measure rumen-protected choline bioavailability. Are you familiar with this technique for determining rumen-protected choline bioavailability, and what are your thoughts about it?
Yeah, I mean, I've read a couple abstracts on this. I mean, I don't have so much of a problem when it comes down to the rumen protection part. When you look at something in a rumen and you see how much is protected at different time intervals, I have a problem with taking a bag and inserting it into the duodenum and looking at what's in the feces. I think that is a tool. I think it is an important tool to help us better understand intestinal release across the GI tract, that complete technique. But even when we ask the question, that is not, I've seen it defined as bioavailability. It'd be very clear that is just not bioavailability and what that represents. I'd call that a measurement of sort of digestible choline. It is certainly not metabolizable choline, and it is not bioavailable choline.
Thanks. So the second question is about that nice table that you show that your PhD student put together, looking at the effect on different metabolites of administration of choline. So you already said that during your presentation that stage of lactation can affect the extent of response in terms of these metabolites. What will be your suggestion on best stage of lactation to use for bioavailability studies on dairy cow?
Well, I'm probably going to step back a little bit on that. I just don't think there's the right experiment that's been done out there to determine whether or not you get more bioavailability in one stage over another. I think you have to do measurements across stage of lactation. I think you're very likely to have differences in the metabolite response. We have some indication of that. I want to point out that at least in the 2 studies that we have done, we call it early lactation, but it's pretty darn close to peak milk production. It's not really like a fresh cow compared to a late lactation cow. I think you see remarkable differences between a dry cow and a lactating cow, that's for sure. There are very stark differences in that choline metabolite response. But it's a little harder so far, just study to study being able to say conclusively whether that response would be similar across stage of lactations or not. I just can't say. You need a study where you test that in a single study or a couple studies to be able to draw that conclusion. Now, if you had to pick one, where would you start? I obviously would want to start with the early lactation cow. Maybe it's a little more challenging to measure sort of where it goes, but I'm not so convinced just yet. And we needed to focus on the early lactation cow because that's the cow that's got the most concerns and the most focus, and we want to set her up for a good lactation. So that's where I would put my attention.
Thanks. So the next question is, so how accurate are the various in vitro tests that's being used by some choline companies to claim bioavailability for their products, in your opinion?
Well, if they're claiming bioavailability, that's inaccurate. I'll just say that. Just don't call it bioavailability, then that's step one. Step two, like I said before, any in vitro approach that attempts to quantify apparent intestinal digestibility, they're overestimating it. They're overestimating what is potentially metabolizable. And I think that's where we need to get to. We got to get to a point where we can define metabolizable choline requirements. We're not there yet. But we got to be very careful about how we use those terminologies. But so for right now, they're overestimating what's metabolizable.
Okay, next question. Has your research given you any insight into what the choline requirements for daily cattle might be? Yeah. Does stage of lactation have different choline requirements? And I think you answered already the second part.
Yeah, I suspect that the requirements are greater during early lactation for sure. It most certainly follows the lactation curve. I think that there's so we in our United States here, we just had our nutrient requirements just published, and there's just the final sentence in that section. It reads something that because of the inconsistencies across sort of studies in terms of what's being reported in terms of either rumen protection or bioavailability or what's metabolizable, they can't come to a conclusive decision about whether it is an essential nutrient. I believe wholeheartedly that it is an essential nutrient. You need science to prove that, not just my word. But you got to look at the good thing for us is if you look at the human side, they've established it as an essential nutrient. When you look at maternal nutrition now, there's certainly recommendations to ensure that there's adequate choline chloride and choline in the diet. I find it hard to believe that that would not be the case for the cow. And so what we need to do is probably moving forward, we need to be more transparent about how these different rumen protected choline products behave in terms of rumen protection, even if it is in vitro test, being more transparent about that, measuring what ends up in feces. More data we can put in the literature, the better. And so we can better compare across studies to just really answer that question for you.
Okay. So next question. Even though rumen in situ studies have their limitation, they can help to estimate rumen protection of rumen protected choline products. Based on your bolus studies, can you estimate the residence time of rumen protected choline products in the rumen?
Yeah, that's a good question. We have a lot of debate about this one because we don't know how fast it ends up in the liquid fraction, but I would say that you don't see the plasma response. The earliest you would see it is probably 10 hours in terms of a plasma response. And so it's certainly, if I asked what would that peak response be, if we were just looking at our data, that peak response in plasma is probably 14 or 16 hours. And so you probably start having entry of these products into the intestine in that 8 to 10 hour window. That's sort of where I'm at. It probably doesn't peak until that 12 to 14 hour window. Really, these are just best guess estimates right now, but yeah.
So the next question is about your opinion on application on farm. You don't have this issue most of the time in US, but in Europe we have small farms. So the question is, what would be the best feeding strategy of rumen-protected choline if you can only feed it during the dry period? Feed more of it during the last 3 weeks, or feed the entire dry period, in your opinion?
And feed, just so I want to get this right, feed more during the close-up or feed less throughout the dry period?
Or feeding the whole dry period, yeah.
The same amount, though, the same amount.
Yes.
I mean, I think we can say that feeding it during the dry period counts. It matters. It's going to improve performance. I can say that. I can say that feeding it in the close-up period will enhance performance. It will, throughout lactation, improve transition cow health. I'm a firm believer in that. Now, if you're asking me the difference between starting at, I don't know, minus 60 or minus 21 days, or approximately 2 months or 1 month, I don't think I can conclusively say if it's superior or not. I would think that, I can't answer that question. I can just say what I just said, that yes, it belongs in dry cow diets. It belongs in close-up diets. Do you get any added benefit throughout the whole dry period? I don't think there's adequate research to answer that question. I'm trying to use science to base to create.
Yeah, sure. Yeah, so sometimes it's just the easy way to apply the product.
Yeah,
but
I'm not going to assume.
Okay. Most people believe that all choline products are equal in performance. Have you seen differences in bioavailability with different rumen-protected products?
Yeah, if you're going to define bioavailability as sort of the choline or choline metabolite response in plasma or milk, we've done studies, independent studies, multiple studies that with a variety of sort of sample sets. And yes, there is a wide breadth of variety. There's no question about that. Absolutely.
So there is a question from Germany. What about choline and lipidoses of the liver?
Okay, so, you look at the totality of the research. There is a general agreement that there's improved liver function with rumen-protected choline feeding. Okay, I would say that the majority of those studies, and this is empirical, mind you, and I'm going to be transparent, that there's an enhancement of triglyceride disposal and a reduction in sort of ketogenesis with choline feeding. There are some studies where you don't see a triglyceride lowering response. I think this probably comes down to either feeding level of the supplement or what was actually bioavailable in that supplement. I mean, if you were to do, if I had to hypothesize that if I had a bunch of rumen-protected choline products that had very high bioavailability, I'm just saying, let's say, 70% plus, something really unique, they all should lower liver triglycerides from the liver. I mean, there's in vitro data to support that too. We actually did a study where we isolated the lipoproteins. I got to eventually publish this at one point. We isolated the VLDL fraction from cows that got increasing doses of rumen-protected choline, and we saw more triglyceride in the VLDL fraction, and liver triglyceride deposition went down with increasing dose.
Thanks. There is another question. Do you have any experience with Biocolony versus rumen-protected choline? I don't know if you're aware of what Biocolony is.
Yeah, I'm not aware. Tell me what
Biocolony is a sort of natural source of choline. It's a mix of herbs, including different choline sources, basically.
So it's not protected?
Not that I'm aware.
Yeah, so that's something new to me. Hey, I learned something about choline today. I'll have to look into that. Now, I will say that, I don't want to rule out the fact that not knowing anything about that product, I think there's actually a little bit of data emerging. I just read a paper from University of Florida that was looking at the effects of choline on fiber digestibility. I don't recall the outcome of that paper, but there has to be effects, I would think, on choline on sort of nutrient digestibility that we just don't appreciate or know yet. So I'm not going to say that there's probably not benefit of unprotected choline, but I can't define what that is. just yet.
Okay.
It's interesting.
Next question is, how is choline absorbed from the small intestine?
Yeah, there are choline transporters. I think I just remembered one. I know the abbreviation for it, like sodium dependent SCLT1 and 2. They're different isoforms. And things that I know about that is that they're responsive to choline. So is choline bioavailability or availability goes up, their expression goes up. That these transporters are more present in the small intestine than the large intestine. But in terms of the cow, oh man, we don't have a clue. In terms of these transporters, that would be an exciting thing to try to figure out.
Thanks. Another question from Italy. Choline has even antioxidant effect. Do you suggest to use it around the peak of lactation during heat stress season?
Yeah, so the heat stress stuff's, I know you asked me a year ago, I probably would have been like, I don't know. Wasn't a believer quite yet. You can say that. I was really, maybe I'm really cautious when I will believe something, and I'm going to really look at the totality of studies. But there is a building body of literature to suggest that choline has benefits in heat stress animals, that it's probably going to protect against oxidative stress. And the more studies that come out like that, the more you believe it. And I'm becoming a believer that, yeah, there is a benefit there that we need to explore more of, especially with regards to immune function. I think it's not just the antioxidant part, or I think it's probably benefits on the immune function that we just haven't even thought about yet.
Thanks. So the last question, because we are already over time, it's recognizing there is currently no choline requirement established yet for dairy cows. Do you have any thoughts regarding choline's benefit as a methyl donor in dairy cows, similar to how the poultry industry uses choline once the choline requirement has been satisfied?
If you're asking me I think I'm trying to understand the question. If you're asking me, is choline a methyl donor, and what are those requirements? Is that what you're asking?
No, I think that the question. So in dairy cow, there is still not any requirement fixed. I mean, even the new NRC is not giving a required doses. So is there any extra benefit from choline as a methyl donor in dairy cow that can be comparable to the poultry industry, how the poultry industry use choline?
Yeah.
Once the requirements are already satisfied, let's say.
Got it. Well, you're really going out there, because first we have to define requirements, and then we have to figure out, okay, if there's added benefit beyond that. I believe that there are requirements, and it would be interesting. It'd be interesting to see how. I think there's going to be some interesting data coming in the pipeline looking at that exact question, because I'll bring it back to this. I know there's been some studies looking at different rumen protected choline feeding levels. Like there was one that was done by one of the papers that you cited that went above that 13 grams, and they kept seeing added benefit on milk production, but also on liver function. And I think and I believe there's probably going to be some work presented this year at ADSA as well, looking at milk performance. And my general indication that there's probably additional benefits on health beyond milk production, right? Because we define requirements often just by the amount of a specific nutrient to support milk production, but you can define requirements to support health too, right? And so you could have a scenario where, yes, you are meeting the requirements to make milk, but then the animal gets exposed to, pathogen, they get sick, and then there's a new set of requirements to try to achieve. And so I think that probably will reveal itself as a possibility. Hopefully I answered that question right. That was a tough.
I think so, yeah. So thank you very much, Dr. McFadden, and thank you everyone for attending today's webinar.
If you have additional questions, please submit them to anh.marketing@balkan.com. The next webinar will be held on April the 6th, when we welcome Dr. Heather White from Wisconsin University presenting Transition Cow Nutrition, Harnessing Long-Term Benefits from Short-Term Choline Supplementation. On behalf of Balkan and Dr. McFadden, thank you for joining us today, and I hope I will see you back for the next webinar. Thank you again, Joe.
Yep, thank you.
Have a great day.
Bye.